Hi Jamie, how are you?
Hi, Catalina. I am well, thank you. How are you today?
Very well, thank you.
Fantastic. Okay. You know, I'm going to share the room because I keep forgetting Clubhouse can do that. There we go.
Good morning everyone, thank you for coming. We'll start in around 7 minutes. Thank you.
Good morning, everybody. Thank you for joining us. Or good afternoon for me if you happen to be of the European or otherwise.
Yeah, today's topic, as you can see by the description on the top, will be that Dr. Edward Morrisey, they discovered a new type of lung cell in the human. Yeah, Dr. Edward, he is at the Perelman School of Medicine, University of Pennsylvania, and he will be here any minute now, and we'll talk about this, this amazing paper that he just published.
It is amazing. A new part of the lung we didn't know about. Well, it makes you wonder what else is around there that we don't know. We'll have to see what the implications are for something like that. A lot of people with these lung problems, and it's definitely referring to COPD. I know several people that have got that problem. So I'm quite looking forward to hearing this talk.
Yeah, it still amazes me that in the last years we still discover new parts of our body that we didn't discover yet.
It's exciting and a little bit scary. There's things happening in there we still don't know. We always have this impression that we've totally cracked the human body, that we know all there is to know about it.
Yeah, apparently, no, we didn't. We think
Well,
that, and then we don't, which is, yeah.
but it's hard to know what we don't know, right? You know?
Yep, exactly. Just assuming that we know a lot is probably the wrong thing. That's all I can say.
It's true. It's true.
Just
Every single time we seem to get a bit overconfident with, yes, we know this perfectly. Boom, something comes along,
exactly.
which is really great though. I am glad that we don't get sort of cemented in our own certainty.
Yeah, that's important to not be that, like, yeah.
Yeah, keep on working on that neuroplasticity constantly.
Exactly. Exactly. Keep learning.
Let me see. So, everyone, take a chance to have a look at the paper. Well, we've got a few minutes. It's definitely an interesting read. Never noticed that, Katerina. This little thing you've got here, red apple, and that's health. Is that the little
Yeah,
emoji topic thing?
it's the topic. It's not my choice. Like the symbols. The symbols are, you know.
Just I wonder who came up with that. Like, think of health. Apple. Yes, that was my first thought when I think of health. Ah, that must be apple a day keeps the doctor away. That's probably where that comes from.
Yeah,
You've
yeah,
heard,
that's true. Yeah.
yeah, that's probably where that comes from. Bit of a twisty, windy mental road they did though to kind of to get there though. Health and apple, but hey, they got there. Um,
Exactly. It's the same engineering and pizza.
uh,
Also,
and okay, really?
yeah, it's a pizza slice. The engineering.
I always wondered about what that pizza slice was doing there. Is this because it's a marvel of engineering or something like that?
I don't, maybe because you do so many nights and order pizza. I have no idea. I don't know.
Or I don't know, maybe because it's circular and it's like the wheel in engineering. I don't know, tell us what you think in the room chat. Why do
Yes.
you think
Why do you think?
pizza
Yeah.
is
It's
connected with engineering?
exactly. It's funny.
Oh, this could always be cause for our own separate rooms, a different time, a different place. What does this symbol signify? And I see there's a little microbe thing, that's the COVID stuff. Was this you picking one that was related to the lungs, Katerina?
Yeah.
I, I, I see how you got there.
Yeah. Of course, everything we can discover about the lung. Hi, Edward. How are you? Welcome.
Welcome, Dr.
Thank you for
Morrisey.
coming. The unmute button, if you're on a iPhone or iPad, should be all the way on the right bottom corner. There's a little microphone symbol. If you press on that, we can hear you. Can you hear us?
Can you hear me
Yes.
now?
Oh,
Yes,
there we
now we
go.
can hear
Okay,
you.
sorry. I'm actually on my computer, so I couldn't find the little button. But
Oh, yeah, I'm sorry.
no, no worries, no worries.
Very, you're very
Thank you so
welcome,
much.
Dr. Morrisey.
Welcome.
Thank you.
So yeah, you'll have to guide me a little bit about how you want to do this. Tell me how you want to go about this.
Sure. So usually, I would introduce you to the audience with giving a little bit, of information about you. Uh, Jamie usually asks like a more general question, how you became to work in this field, and then we would keep it relatively short because I understand you have 1 hour, right?
Right. Yeah, I have another meeting
Yeah,
to attend.
exactly. So we'll keep it right in that time, and then if you would like to give a summary about the work and how you came about it, that would be wonderful. And then if we still have some time for a few questions, that would be that would be great. So
Sure.
perfect. So yeah, let's just start. Welcome everyone to the Science Society. We are very honored to have our guest speaker here today. and talking about his work. And let me give you a little bit of information. Dr. Edward Morrisey, he is at the Robinette Foundation Professor of Cardiovascular Medicine. He's the scientific director on the Institute for Regenerative Medicine at University of Pennsylvania and director of the Penn Center for Pulmonary Biology at the University of Pennsylvania. And he got his bachelor in science in microbiology from the University of Illinois, and his PhD in molecular biology at the Northwestern University. And the overarching goal of his lab is to understand the cellular and molecular processes required to generate, maintain, and regenerate by different tissues with in the mature or adult mammalian respiratory system. And yeah, thank you so much for being here. And Jamie, the stage is yours to ask a couple of questions.
Thank you very much, Katharina. So, first of all, Science Society definitely welcomes you, Dr. Morrisey. You're working on some very exciting stuff. But we'll start it off by taking you way back to your origins, because we actually love hearing what got someone into science in the first place. First of all, can you tell us what was it that sort of started you on this kind of field of academia and search for knowledge?
Okay, great. Well, it's wonderful to be here and to be talking to you today. So, I grew up in a very small town in the Midwest of the United States, and I always was, intrigued by just nature, everything around me, whether it was, insects or looking up at the stars and such. So I had a sort of an ongoing, love with just the world and nature itself. So when I went to college, I decided I wanted to study microbiology, the study of microorganisms. I had this fantasy because I wanted to sort of combine my love of nature and my love of space into to sort of understanding exobiology. And there actually is an office. I think it's called something different now, but there was an office at NASA. There was the Department of Exobiology, and that was the study of potentially if we ever got to Mars or something like that to understand what microorganisms will do. So that's sort of like that sort of cross between almost like science fiction and science was really what inspired me as a young person. So I studied microbiology in college. I actually went to work at Abbott Laboratories for 4 years before I went back to graduate school. It was a really important thing for me to sort of get out and see what happened when you try to commercialize science. Went back to graduate school, and then did a postdoctoral fellowship at University of Chicago, and that's where I started really, examining, the development of the cardiovascular system and the pulmonary system. And then after that, I uh, uh, got a faculty position here at UPenn, and really, sort of just moved on and trying to understand primarily in the last 5 to 10 years, really how lungs develop and how they respond to external stimuli, and in the case of many of the things we're doing now, how they respond to injury. And of course, that's obviously quite topical in the in the era of the COVID pandemic, because it's predicted that having this widely infectious disease infecting probably maybe ultimately billions of people throughout the globe, there's going to be a huge increase in chronic lung disease, in addition to obviously the acute symptoms of the pandemic itself. So there's a lot of interest in how the lungs respond to injury, how they regenerate, and they are a regenerative organ, in comparison to, say, organs such as the heart, which does not really readily regenerate in mammals. So that's really sort of how I got started. It's a very quick bio of myself, and certainly open to other questions.
Even the quick bio of yours is absolutely fascinating, and I bet you didn't expect what you're working on. I mean, yeah, it always had big relevance, but with the COVID pandemic, it probably brought the relevance into the stratosphere at that point, right? Um,
Yes.
what what was it that got you into the lungs then specifically? what was it that kind of moved you in this direction? Because as I heard your path, it sounded like it was quite steady, but there must have been something that you thought, lungs, breathing, let's go there.
Yeah, so like I said, I started out working really primarily in cardiovascular development as a postdoctoral fellowship at the University of Chicago, and we were working on some transcription factors called GATA, GATA transcription factors. It's a family of 6 transcriptional regulators. They're known as pioneer factors, or some of the primordial transcription factors that sort of insert themselves and help open up DNA for genes to be expressed. And by doing that, one of the gene knockouts, one of the knockout mice that we generated, actually had a lung defect, and it was not expected. But when I looked at that, I was just really enamored by just, in some ways, the beauty of the respiratory system. I mean, it's this very delicate and elegant looking branched network of airways that has a parallel branch network of blood vessels that obviously are required for gas exchange. And there wasn't really that much known at the time about lung development, or about how lungs interact with the external environment, how they're injured and how they respond and regenerate. So it really was a wide open field. So some ways it was pragmatic. I was in a field that it was, a very large and very intense scientific field, cardiovascular development and biology. And I saw this opportunity to study something that really was underserved in many ways.
That is definitely fascinating, because I know that a lot of us laymen, we just think, oh, the lungs, you breathe in, there's air, you breathe out, there's, you know, carbon dioxide. There we go, it's already solved. Um, it's quite enlightening to see how much we didn't know. Anyway, thank you
Yes.
very much for the questions, and are you ready for your talk? The floor is yours, Doctor.
Okay. So I thought I would just, sort of go through the paper fairly quickly in 10, 15 minutes at most. And certainly interrupt me at any point if you have questions. So I think everyone has the copy of the paper in front of them. Is that true?
Yeah, we shared
Yeah.
the link, and yeah,
Good.
everyone has it.
Good.
Thank you.
No problem. So the genesis of why we asked the question that this paper addresses really comes down to the fact that, as we were just talking about, I've had a long-standing interest in understanding how the respiratory system develops and how it responds to the external environment, including diseases, acute diseases, or a chronic lung disease. So with that in mind, we use the mouse as our workhorse. It's the model system that most labs working in respiratory biology use, because it's tractable, it's easy to use. We can do nice genetic gain and loss of function experiments with it. And the lungs in the mouse are very similar to the lungs in the human, but they're not identical. And that was the key. That was sort of the inflection point, that we were trying to understand what makes the human lung different. Obviously, it's huge. The entire surface area in the human lung is about the same surface area that's on a tennis court. So it's got this enormous interaction between the external environment that is folded up inside of your chest, almost like in tissue origami. And obviously, that's very different than a little mouse that has a much smaller, multiple orders of magnitude smaller respiratory system. So that was the overarching question. So what we did to get at that question is we utilized what is a very common technique now. It wasn't so common 5 or 7 years ago, but a very common technique of using single cell RNA seq of the human lung, a normal human lung, as well as diseased human lung. And the disease we looked at in this story is chronic obstructive pulmonary disease, or COPD, which is the most common lung disease in the world. And we'll get back to that in just a moment, and the importance of that. So we did this single cell RNA-seq analysis, and what that does is essentially it examines the transcriptome, or the gene expression in every single cell, and you look at this over thousands and thousands, tens of thousands of cells across multiple different donor patient samples. And in Figure 1 is really the sort of gestalt of that first part of the story. 1A shows you in a diagrammatic fashion sort of the differences between the human and the mouse lung. And I'll bring your attention to the region that's shown in orange in the human lung, and that's the respiratory bronchioles, the respiratory airway system. And that does not exist in the mouse. So this is a structure that doesn't exist in mice and is unique to humans and other large mammals. So that's where we really wanted to focus. And then we wanted to ask, well, are there unique cell types in that region? And indeed, through and I don't want to go into too much detail, I'll certainly answer the questions that might come out of this. Through a lot of analysis, we identified a cell type that we just called it a respiratory airway secretory cell. So secretory cells are found in airways of human and mice and other mammals, and they, as the name suggests, they secrete mucins and other sort of molecules that help lubricate the large airways of the lung and keep it open and keep it humidified and helps clear it also of particulate matter. So this is a unique cell that we did not see in the mouse. It had a different gene expression pattern than in the mouse. We also only saw this cell in the very distal airway. So if you look at, say, D and G in figure 1, D is the distal airways, and including the alveolar space where the gas exchange occurs. And G is the sort of UMAP projection of the proximal airways. And the cells we're talking about here are in that sort of bright pink. So they're observed in the distal airways, but not in the proximal airways. So this is sort of interesting, right? So this is a cell that is found in this respiratory bronchial region, respiratory airway region, a unique structure in the human lung, and it's really only found in that region of the human lung as well. Over, on the bottom right of figure 1, H through M, is immunohistochemistry looking at different marker genes of this respiratory airway secretory cell, or RAS cell, as we call it, as abbreviation. And one of the hallmark genes for this cell type is this gene SCGB3A2. And this is sort of what we actually use to identify the cell. We've also used it to track the cell in tissue samples. We looked at human, obviously, and I think H shows you this sort of longitudinal landscape, low magnification shot, how those red cells are found in the RB region, respiratory bronchial region, not really found so much in the terminal bronchial on the far left. And they're also not found deep into the alveolus, or the alveolar region where the gas exchange is occurring. So they're found in this very sort of narrow window, this respiratory bronchial region, and not found in other parts of the lung. The rest of this just shows like that these cells are unique and distinct in other little, sort of higher magnification shots. Sometimes, as you see in K and J, you will see these cells express this gene SCGB3A2, which again is the hallmark gene for RAS cells. Sometimes they'll also express genes for the alveolar type 2 cell, whether that's SFTPC or LAMP3. And I should take a step back here for a moment to explain some of the cells in this region besides the RAS cell. So the most important cells for the rest of the talk will really be the cells in the alveolar region, which are called alveolar type 1 or alveolar type 2 cells. Alveolar type 2 cells are the cells that generate the pulmonary surfactant in the lung. Again, this is the part of the fluid that keeps the lungs from collapsing every time you take a breath. It lowers the surface tension, allows you to open and close your airways and your alveoli without it closing permanently and not, allowing it to reopen. So they're critical. Alveolar type 2 cells are critical for making pulmonary surfactant. They're also critical because they act and harbor within their population the stem progenitor activity in the alveolus. In other words, they have the ability to, in response to injury or chronic disease, proliferate, make more of themselves, and also make alveolar type 1 cells. So alveolar type 1 cells are this very thin, very large, extended cell that covers about 90-95% of the alveolar surface area. It actually forms the gas diffusible interface with the vasculature, the pulmonary vasculature, capillary distal vasculature in the lung. So it is critical for exchanging gases because it's got these very thin cellular processes that overlies the vasculature in the alveolar space. So that's just to let you know, those two cell types, alveolar type 1 and alveolar type 2, they're critical for the function of the alveolus, which is to exchange gas. And going back to the RAS cells, sometimes we saw a very small minority of those RAS cells expressing marker genes that were common to the alveolar type 2 cell, this progenitor cell in the alveolus of the lung. And then finally, in I, L, and M, is really taking a look again at this RAS cell marker, SCGB3A2. And we looked at this in ferret as well. So ferrets are an interesting animal model. They have a lung that is more similar to humans than mouse does. So it was utilized in this context to say these 3A2, SGB3A2 positive cells also seem to exist in this unique structure in the ferret lung that is very similar to the human lung, so the respiratory bronchiole. So this whole figure really is designed to say we looked deeply using single cell RNA transcriptomics. We found a gene signature and potentially a unique cell type that's in the human lung but does not exist in the mouse lung. It also seems to exist in the ferret. So that's figure one. Did anybody have any questions before I go on to the next figure?
I think I'm good, but Jamie, do you have a
Uh,
question?
Actually, I did have a quick one. When you talked about is it the alveolar type ones? Are these the ones that regenerate? Yeah, if there's damage, it knows to regenerate if it finds damage. Is
So
that what you were?
yeah, so all of these cells can regenerate after acute damage, right? So we'll talk a little bit about the three SGB3A2, the RAS cells, in a moment. But say after an acute damage, after like influenza injury, which is a model that we use in mouse, we can infect mice with flu A, and they could get very very sick, and they get significant damage to the lungs. In that context, what happens is the alveolar type 2 cell will start to proliferate because a lot of the type 2 cells and type 1 cells after influenza infection get killed off. So the alveolar type 2, the remaining cells will proliferate, they'll grow, and then some of them will differentiate back into type 1 cells. So that's sort of the response to an acute infection by type 1 and type
Right,
2.
right. Because I'd like to. This can wait for the end, but I wanted to ask. I'll put the question down here now. Is how do the cells know? Like, does it get a message going? We're healthy, do nothing. We're healthy, do nothing. Oh, we're damaged. Start making more, please.
It's a great
Um,
question. Yeah, and maybe actually towards the end,
yep,
I'll try to go
yep,
through the
absolutely.
figures, but it's a really good question. And the short answer is we don't really have a good idea, but there are some hints, some ideas about how that occurs.
Excellent. Thank you.
Okay, so I think the other figures we'll probably be able to go through them a little bit quicker because that first figure is really a lot of data, and there's a ton of supplemental figures that was really just to establish the identification of these cells. So we have these cells. So the other conundrum we have when we work in something that is unique to the human system is that we don't have obviously the obvious ability to use mice. And as I said before, mice are an incredibly wonderful model organism. We have all the genetics, we have the ability to do these beautiful genetic gain and loss of function experiments, gene knockouts, gene overexpression, the ability to do fate mapping, the ability to tag cells using Cree-Lox recombination, and then ask what happens to what you just said. That what happens to the response of these cells and track the response of these cells after an acute injury. So as Jamie was just asking, it's like we can do that in mice. In humans, it's much more difficult because we don't have those elegant genetic tools to do that. But what we do have in the human system are stem cell models and organoid models. So figure two now that we're going to look at is how we built a stem cell / organoid model to understand the abilities of these FCGB3A2 RAS cells and whether those cells could self-renew, make more of themselves, but also turn into this alveolar type 2 cells. I showed you in the previous figure, sometimes we would see these RAS cells express markers of the alveolar type 2 cell. And I didn't go into deep discussion about the transcriptomic analysis, but the transcriptomic analysis using something called trajectory analysis suggested a relationship between the 3A2 RAS cells and the SFTPC AT2 cells. So informatically, as well as histologically, it looked like there was a relationship between these two cells. And one thought was that maybe the RAS cells, whether it's a normal homeostatic turnover or in response to an injury, maybe they can make an alveolar type 2 cell. So we developed this, what we thought was pretty elegant bilineage, dual lineage reporter stem cell model. And in this context, we inserted, using homologous recombination, we inserted a cherry red fluorescent reporter into the 3A2 human locus and human ES cell model. And then we also inserted a green EGFP fluorescent cDNA into the SFTPC human gene. So both of these reporters were inserted into the RUES2 ESL model. And we have protocols to turn these cells into either RAS cells or into alveolar type 2 cells from previous work that we've had with our collaborators. So we took these cells, right, and we turned them into RAS cells. And that's the red state, as you see in A. So these cells will turn red, they'll grow, and you can passage them, several times. They'll grow in these nice little sphere-like organoids. What we found is that when you took the red cells and you simply put them in media that help promote them to turn into AT2 cells, within 2 to 3 days, they would rapidly turn off markers for the RAS cell state and quickly turn on markers and gene expression programs for the alveolar AT2 cell state. This is really rapid. Normally, if we were to try to take these ES cells and make alveolar type 2 cells, AT2 cells, normally that would take 2 to 3 weeks. But when they were already in the red RAS cell state, they would turn into AT2 cells within 2 to 3 days. So it was an incredibly rapid response. In B of this figure, what we then showed also was that we could take the red cells, the RAS cells, we grow them, passage them, put them into the media that would turn them into AT2 cells, turn green on, turn red off. And then we could continue to passage those green cells as well, multiple times. But when we tried to take those green cells and put them back into the media that would promote the RAS cell, the red cell state, they would not go backwards. In other words, they could go from red RAS state to green AT2 state, but they could not go from green AT2 state back to the RAS red state. So that suggested to us that it was a unidirectional differentiation process, that RAS cells could make AT2s, but AT2s could not make RAS cells. And through a series of experiments, I don't want to go into too much detail, we found that a couple of molecular pathways seemed to govern this process. They were Notch signaling, which is important for cell-cell, direct cell-cell communication, helps to maintain, we know it helps to maintain airway cell fate in the mouse. And indeed, these RAS cells, it seems to maintain and promote their RAS cell state. And it seems to inhibit their ability to turn into AT2 cells. So if you inhibit Notch, DAPT is the small molecule that actually inhibits the Notch pathway. If you inhibit Notch, over in C, you can see the RAS cells just turn off SCGB3A2, which is shown by loss of the red fluorescent marker, but it actually increases the intensity of the green cell marker, the alveolar type 2 cell state. So it seems to be important to maintain the RAS state, and inhibiting it will actually sort of push the cells into the AT2 state. Conversely, over in E, we found the Wnt signaling, and we know from multiple different experiments from our lab and other labs that Wnt signaling is important for the AT2 state. It is not important for the 3A2 RAS cell state. And indeed, if we put an activator Wnt, KIR, which is a GSK3 beta inhibitor, activates the Wnt pathway. We put that into these cultures. It shuts down 3A2 expression, red fluorescence, and it promotes and turns on SFTPC, the alveolar type 2 cell state. So it looks like Notch and Wnt are sort of balancing the ability of these RAS cells to either self-renew and stay in the airway, or to turn into alveolar type 2 cells into the alveolus. And I'll pause there for another moment and see if there's other questions.
Yeah, thank you. For me, I'm fine. Jamie, Katie, do you have a question right there?
For now, I'm happy to wait till the end, but thank you.
Okay, thank you.
Cool. All right. So the next figure, figure 3, is a pretty simple one because it's sort of a repeat of figure 2, except we did this with primary cells isolated from human lungs. So, one of the questions we got was, okay, these stem cell models, as I just showed you in figure 2, they're great, they're wonderful, they're elegant. Again, we can put in fluorescent reporters and track cells as they turn, go from one state to the next. But is this really how, in this case, the SGB3A2 RAS cells, is this really how they behave in vivo? And again, we certainly can't do lineage tracing in humans. That's obviously something that's just not possible. But if we could isolate the primary cells, those RAS cells from the human lung, we could still do organoids. We could culture them in 3D matrices and say, okay, do these, RAS cells derived from the human lung, do they actually turn into alveolar type 2 cells as denoted by SFTPC expression? And indeed, we were, using bioinformatics, we're able to look for and find a cell surface marker for RAS cells that was, we could enrich for those cells. It's called CCAM6 or CD66C is the other name for it. So we could use FACS analysis, basically antibodies, to pull these cells out. This marker is expressed in other cell types in the lung, so we sort of negatively gated, got rid of all the other cells using other antibodies. So we pulled out a population of cells that was, CCAM6 positive, but negative for alveolar type 2 cells, negative for basal cells, the other population that expresses this marker. And through a series of experiments, we showed that, what we pulled out was pretty pure, this RASC population. So when we put that then, those SCGB3A2 positive cells into an organoid model system and look at these after 3 weeks, what we found is about 65% of these organoids were alveolar type 2 cells. So they started out completely negative for alveolar type 2 markers. But within 3 weeks, essentially almost 2/3 of them would turn into alveolar type 2 cell organoids. Again, suggesting that these cells have the ability to turn into these alveolar type 2 cells, another progenitor cell. So it's looking like these RAS cells at this point are a progenitor that is capable of generating another stem progenitor cell in the lung, and where it's located at the very distal end of the airway, suggesting it's probably helping to maintain these cells in these multiple different compartments in that unique respiratory bronchial region of the human lung. So that's what this figure allowed us to do, was to say, okay, the actual cells themselves from a human lung have this capability. I'll pause there for a second. Does anybody else have any questions before I go on to the next figure?
I actually do, but I really want to make sure you get through your talking time. I know we are being limited, so I will hold on to mine.
Okay.
End, please.
All right, I'll just go on to the last figure then, the last main figure in the paper, and certainly I'm happy to answer any questions with this data as well as what's in the supplements. So the last figure then, gets to the point where we've identified a new cell type that's unique to the human lung, doesn't seem to be found in the mouse lung. It's in the ferret lung, because ferrets have this respiratory bronchial structure, and their lungs are more similar to humans. So then we wanted to say, if this unique cell type has the ability to populate both this respiratory bronchial region and also make alveolar type 2 cells, this region I should actually discuss for a moment in the context of disease and injury. This respiratory bronchial region has been known for many years to be a highly susceptible site of injury and chronic lung disease. Using imaging, CT scans, etc., longitudinally in the human population of people that have different pack years of smoking history, have COPD, it's been known that this region is disrupted way before people actually have other phenotypes of lung disease like COPD or emphysema. So it seems like disruption of this respiratory bronchial precedes many other symptoms and phenotypes in chronic lung disease. So again, that suggests that this region is important. It might, if you damage this region or this region gets perturbed, it might then itself be causal in having these other downstream phenotypes appear in people. So we wanted to ask, is this RAS cell disrupted in the most common lung disease, which is COPD, chronic obstructive pulmonary disease? So we started out with single cell transcriptomics on a group of COPD samples. And what we found right away was that we saw this new population. So we saw the RAS population, again, that's in dark red and A. But then within the AT2 population, which is in the blue colors, we found that there's this new subpopulation that did not really come out in the normal lung. And that's this new population is a population of alveolar type 2 cells that has increased expression of SCGB3A2, the RASC marker. And again, suggesting that maybe as these cells are going from the RASC cell state into the alveolar type 2 cell state, they keep, they have elevated expression of RASC cell markers as they do this, and they're acting abnormally. And then, basically through a series of other experiments in B and C, this is all the informatic data. We definitely show that that exists as a unique population, the alveolar type 2 population. And in D, that just shows you the co-expression of these two markers, SCGB3A2 and LAMP3 for alveolar type 2 cells. So there's definitely cells that are expressed, increased numbers of cells expressing markers of both of these cell types. And what was really, really cool, at least for many of us, was when we looked at, if you look at panels E, F, and G, and these are, and H as well, and these are basically going back and looking at people that don't have COPD. Like in D, is frank COPD. E and F and G and H are basically people, as well as ferrets, subjected to increasing amounts of smoking. So in humans, obviously, this is just, we get samples, human lung samples from people that have different, have been smoking for different amounts of time and years. And I think, just basically you can look and see in the graph in G, I think probably illustrates it the best, is that the more you smoke, the more of these double positive sort of confused alveolar type 2 cells that have increased levels of RAS cell signature in them, the more of those cells you see. And looking in the ferret, we could actually do the experiment. We can actually subject ferrets to 6 months of cigarette smoke exposure. We see the same thing. We see the increase of these double positive cells. Now, these double positive cells are messed up in various ways. They have changes in metabolism, changes in immunological response as well, at least by gene signature analysis goes. So they're definitely not normal alveolar type 2 cells, and they suggest that these RAS cells are going to misbehave in some of these chronic lung disease models or chronic smoking injury models. So this really does bring some, sort of disease relevance to the finding of this new cell type, that it's misbehaving in these disease and injury models. So I will pause there and take any of your questions.
Thank you so much. This is such an amazing and very important work. So really appreciate you taking the time and coming here to explain this to us. And that these cells are highly involved in disease is quite interesting. Do you in the future, or maybe you started already, would have kind of a screening, a pre-screening to see if there's predisposition maybe in these cells in people that then end up getting lung cancer versus not, or being more affected by COVID versus healing better? Is there, a plan, or is there data that shows like a predisposition or a vulnerability for those disorders in those cells?
So yeah, great. I'm going to sort of answer that with sort of divide that into two questions, I think, Katerina. So one is our knowledge of this cell type. Can we track the behavior of this cell to sort of predict, either primarily either maybe the response to acute injury or in chronic lung disease? And I would say, that's one of the questions we have. Can we potentially find biomarkers, that say this cell is expanding abnormally or not expanding enough, and then correlate that with, the response of the patient to any of these injuries. So that is certainly something we're very interested in and are looking at. The other is, is really the question of how does this cell behave in different disease models? I talked to you about COPD. It's the most prevalent chronic lung disease, but now that we're sitting here in the world of COVID and an acute, respiratory and infectious disease, how is the cell going to behave, in that model? The weakness we have with humans is everything is observational, so we can't really perturb the system in a controllable way. But we are looking at people and samples from COVID-19 patients. We're also looking at people and samples that people that are deceased because they had lethal influenza. I mean, as much as we talk about COVID, there's still thousands of people a year, tens of thousands of people a year, just in the United States alone, that die from lethal influenza. which is similar, as far as how people die from ARDS, acute respiratory distress syndrome, as COVID-19. So we are definitely looking at the behavior of these cells in those acute injury models. It'd be shocking to me that these cells are not affected, but we really are in early days. And we don't know exactly how yet that they will be affected.
If I may ask a question about speculative therapies that might result from this, could you comment on that?
Right. So I think probably, I mean, for us, myself, and for the first author I should mention, Maria Basil. She's a MD PhD pulmonary fellow in my lab, incredibly talented. We're going to hopefully keep her on as faculty here at University of Pennsylvania. And her clinical interest is COPD. So a couple of statistics that are a little, shocking to me over the last couple of years that I wasn't really aware of until we started working on this is that WHO, World Health Organization, predicted back in I think 2010 or 2011 that COPD would be, the third leading cause of death by 2030. It actually became the third leading cause of death in 2018 or 2019, so way ahead of schedule. And it's now predicted that COPD could become the number one cause of death in the world in the next 10 to 20 years. And it probably will be. And I think these predictions were all made before COVID-19, before we had this enormous impact on our respiratory system. So this one disease alone is likely to become the leading cause of death in the next 10 to 20 years, which 10 years will go by pretty fast. And so from the standpoint of like, how is this going to leverage our ability to develop or new therapies or even just understand disease better? COPD, we don't even really know what COPD is. It's a degenerative disease. It's basically the lungs start to fall apart and it's progressive and there's nothing we can do about it. So I think, we have a whole lot of information we still need to learn about this disease at a cellular level. But our hope is a couple of things I think that we might be able to address is one, can we come up with biomarkers, as I mentioned earlier? The sad fact is something like COPD, people don't show up to the hospital until they're really, really sick. You have a lot of what's called reserve capacity in your respiratory system. You can walk around with 1/3 of your lung function being gone, and you won't even notice. It's not there's so much reserve capacity. So when you start losing lung function, you've already lost almost half of your lung as far as functionality goes. And at that point, the disease has progressed so far that it's hard to reverse it. So if we can come up with biomarkers, maybe that we can test people, say a blood test or something, maybe it's something that's given off by these RAS cells when they're sick and they're abnormal. That I think would be huge because if we can intervene earlier, we have a greater chance of reversing the disease. But obviously, the ultimate goal is to reverse it by reactivating the stem progenitor activity of these cells, and obviously multiple cells in the lung. It's a very heterogeneous organ. So I think right now we're just in the mode of trying to acquire as much information as we can because we still don't understand some of the basic elemental facts of some of these diseases.
Wow, that sounds like quite a tough problem, but lots of potential for, I think, early interventions. As you said, I myself work on rapid sensor ID, and we deal with biomarkers, so it's always something that we're on the lookout for, especially given the new, I guess, generation of electronic equipment that kind of miniaturizes this kind of point of care potentially, so that folks could make these kind of detections earlier at home. I myself had a problem with delayed diagnosis with my cancer. It delayed my treatment by about 10 months, having argued with 7 physicians who weren't familiar with my particular disease. And and that's me as a biophysicist. So I can only imagine the kind of difficulty, even if people do have some disease and it's obscure, or in this case, very difficult, I think, to diagnose, and perhaps people don't even notice that they have the problem. So it seems quite vicious. Nature is so cruel sometimes. Thank you for the answer.
I wanted to ask, so is the increase in that disorder related to more toxins and pollutants that we are exposed to? Is that related? And maybe there could be an indicator of just general inflammation in the body related to responses to environmental impact. Would that be a good enough indicator to
Yeah.
pursue? Like,
Yes. So the increase in COPD is due to a couple of things, and one of them is increased pollution from biomass, burning of biomass, and especially in poorer countries. But it's also good to know to understand that about 27% of people with COPD don't smoke and are not subjected to massive levels of biomass or pollution. They're in areas that are not overly polluted. So there's probably an underlying genetic thing, but definitely the environmental insult is a huge contributor and probably the primary contributor to a disease like COPD. So yeah, I mean, I think obviously one of one of the biggest pushes, certainly in here in the States, has been to decrease smoking, right? Because it's still a huge contributor, but it's not the only contributor. Um, and ultimately, I think that there's a lot of people that smoke a lot, smoke a ton, and yet never get COPD. So there's a lot of details we don't really understand. Obviously, these environmental insults increase dramatically the significance of these chronic diseases and other chronic lung diseases beyond COPD as well. So I think that's a huge issue, and we still are trying to figure out what the interplay between the external environment and the lung is.
do you think maybe non-coding DNA that is more transposable elements, maybe even close to more the telomere region, could be a contributor that that elicits this? I'm just starting to think already. What could a good biomarker be? What
Yeah.
would
No,
we
no. Yeah, I think it's a great question. So in COPD, it's unclear. I will say another very common lung disease, idiopathic pulmonary fibrosis, also called IPF, has been known and actually been demonstrated to be primarily caused. A lot of it, at least, is caused by telomerase mutations in the telomerase pathway. So, so that as far as like things being close to the telomeres and losing that telomerase as sort of preservation of genomic information, that is certainly that certainly underscores another fairly common lung disease. And whether that has an effect on COPD or not, it's unclear. No one has actually looked at it carefully, and certainly from the say GWAS screens and stuff like that, that has not come out. But I think again, chronic diseases, whether it's COPD or IPF or other tissues as well, these are generally as we sort of call them degenerative diseases, right? They're tissues in the lung in particular does have the ability to repair itself and maintain itself by certain cell types proliferating and redifferentiating. But something happens that sort of tips that balance, right? Tips that balance to the point where either it's degenerates in a progressive fashion, or even maybe, ages, put aging in quotations there, not like your true aging, but sort of ages, it degenerates faster than your rest of your body or normal life span would indicate. So I do think that some of these things like telomerase mutations, pathways, senescence is another thing that people have been looking at recently. Is there an exaggerated level of senescence in some of these lung diseases, and can we come up with biomarkers for that? So there is definitely a lot of interest in that stuff.
And can I ask, when you were talking before about the alveolar type 2 given, is it double please correct me if I'm making any mistakes here, but the type 2 given double positives, you said, when they were being damaged. Is that messing up whatever signal you said that we're not totally sure what it's doing to say we're damaged, please make more type 1?
Right. Yeah. So that's definitely a great question. We think it might be. We don't have a lot of direct evidence yet because, again, working in the human system has its limitations. But we think that those damaged double positive cells probably don't have as much of an ability to, say, proliferate or respond to a chronic diseased region. I can tell you that they do have changes in their ability to respond to the immune or inflammatory environment. And one of the sort of observations people see all the time in human lung disease patients, chronic lung disease, is there's an increased inflammatory cell, component to these chronic diseases. Obviously, that's going to happen in any sort of acute injury or acute disease, but it's interesting that it happens also in these chronic diseases. And you have to imagine that maybe, what's cause and effect is a little bit unclear, but certainly if these cells, which normally we would think the RAS cells normally could make alveolar type 2 cells, and then those alveolar type 2 cells could make alveolar type 1 cells. If that sequence of events is disrupted because the cells are either responding to and or just intrinsically have some defects related to the inflammatory environment, then yeah, I think that could have a really big impact, and that's something that we're very, very interested in looking at further.
So is this a little bit like the lungs actually have the capability to regenerate and heal, but there's the distress signals not getting through so that they don't know to start repairing things?
Yes, that is exactly right. Lungs, as we've talked about, have a quite expansive ability to repair. I mean, and we've seen this recently with the COVID-19 pandemic. If you look at a CT scan of somebody that comes in, they're in an intensive care unit, they're on a respirator or ventilator, their lungs are just, it's just a mess. I mean, it's just blown up. But these patients, if they survive, come back in 6 months later, and their lungs aren't perfect, but 95% of that tissue damage is been repaired or regenerated, which is quite remarkable because of the intricate and complex structure that the lung has. So there's a lot of ability to repair and regenerate. So I think that it's these chronic diseases, though, where it's like if you're smoking over a period of 20, 30 years, you're giving like micro insults, like micro injuries over, a period of that time that you just sort of wear the system down, right? I mean, it's like the acute injuries happens rapidly within a few weeks, a few months, but the chronic insults that happen over decades, that's a much, much more difficult thing for the tissue to respond to. And at some point, it just wears the system out. The other thing to think about also is, there's probably an epigenetic component to this. It's not like the genes themselves change, but the regulation of those genes, including the epigenetic regulation of those pathways that are involved in tissue repair, are probably altered over time. And once that starts to go downhill, it's probably hard to reverse itself.
Fascinating. And a question from someone in our audience, Victoria, she was asking, do you think your work might lead to regenerative therapies?
We hope so. It'll obviously take some time. I mean, that's the brutal truth is that this is a cell that we didn't know existed a year ago. So it'll take some time once, to understand how the cell behaves and all the disease modeling we talked about. But we do hope that it will. I think, ultimately, there's going to be probably two basic ways that we're going to be able to repair and regenerate the lung. One is to leverage the pathways that exist that Mother Nature has already designed for this. I mean, the thing that's sort of humbling when you do this is that you realize that nature figured this out, thousands, millions of years ago. The ability of the tissue to repair in a coordinated fashion is there. So we just have to figure out how that happens and figure out ways to leverage things and turn things on and off. The other way is to potentially put cells that we grow in a dish back into the lung, transplant them back in. And that's where some of the stem cell models, could potentially down the road have a real impact. And we are working avidly on this within our own lab and working with collaborators as well. Can we grow these cells in a dish, transplant them back in? Will they take up residence in the lung? Will they behave normally? And will they regenerate damaged tissue after that? So there's a lot of hope. And, I think it just requires a lot of brilliant young minds to get in there and do some of the hard work.
That's amazing and exciting. And one question that I had from I'm sorry, please.
Leonard, he
Sorry.
joined the stage. I wanted to check with him, and then I'll get
Absolutely.
back. Thank you. Hi, Leonard. Welcome to the stage. Do you have a question?
Yeah. Thank you so much. I'm just enjoying. I don't have a question related to the lung cell type thing, but my question is it's not related to this, so I'm in a dilemma whether to ask or not to ask it, but it's related to this. It's about the genotype thing because some time back I had my genotype uh checked, and then the doctor said it's an AC. Yeah, I think he said it's AC. And when I googled it, people were saying all kinds of things, and I was kind of discouraged. But later on, I went to the hospital, and then the doctor said there's nothing to be worried about. It's okay. It's fine. So I wanted to ask if you have AC genotype. Is that okay?
So, I don't know, but the short answer is I don't know, Leonard.
Okay.
I think one of the upsides and downsides now is that we can genotype ourselves, to a fairly high resolution. The downside is
Okay.
we don't know what these things really mean, right? Um,
Okay.
it's a real challenge for scientists and physicians in particular, as we learn that maybe some of these genotypes are associated with some disease states, but it's an association. Some of these things might increase your relative risk by a little bit, but overall, it's not going to be that dramatic. So I don't know about this particular genotypic change that you describe, but I think if your doctor doesn't think it's a big deal, you're probably fine. It's my
Oh
best.
yeah, 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 absolutely, absolutely. He said it's not a big deal. He said it's very good because they are very resistant to some minor diseases. So they are very strong. So yeah, but so maybe it could be that what I read online is just maybe somebody's blog and is just trying to put something together, which
Yeah,
which probably isn't true. Yeah, because it wasn't backed by any research or any finding after all.
exactly. Stay with the science and stay with the facts. I mean, people say crazy things online sometimes.
Yeah, yeah, it's true. Absolutely, absolutely.
Thank you.
Thank
And I
you.
Jamie,
think
go
Thank you
ahead.
so much. Yeah,
You-
thanks
I
so
think
much. Yeah.
Thank you.
I think the rooster in the background has a question.
Jamie, you had the last question? Or does anyone have a last question? We have 5 minutes left, so please go ahead. Yeah, Jamie?
Yeah, I did have a question.
Katie?
Just
I'm
I taught
sorry,
myself on. Oh,
Katie,
beg your
did you
pardon.
unmic? You have a question?
Go ahead, Jamie. I'll go after you. Thank you.
Um, I was just going to quickly ask him, how did we miss this cell type for such a long time, and how did you discover it? Because this is a big thing, finding a new cell, or well, not a new cell, but a newly discovered one, a new
Well,
cell type.
yeah, I think it's from advances in technology. Simply put, I mean, we were not able to really profile, tens or hundreds of thousands of cells in the human body up until like the last 5 to 7 years. So, it's really just having the technology to go in and do that is really what it comes down to. We knew this structure existed for quite some time. Anatomists, classic physician scientist anatomists back in the 1950s and 60s had seen in the respiratory bronchioles strange looking cells, but they didn't know what they were. We just have the technology to go in and look at them more carefully. So that's really how it came about.
Thank you so much, Katie.
Hi, it's Katie. Thank you so much, Dr. Morrisey, for such a fascinating conversation. Really, really cool research. Thank you, Katerina, and everyone for hosting this space. Amazing conversation as always. Um, I had many questions. Some of them have somewhat been answered. I'm really interested in the application and potential future application for COVID. And also just in terms of, as io Eric also mentioned, in terms of biomarkers, and someone who is a geneticist and also very interested for personal reasons in COVID. You mentioned epigenetics and biomarkers, and I'm just wondering, without having to give too much away, or even conceptually, are there any sorts of biomarkers, firstly, to assess risk? I'm interested again in the epigenetics, and then if there's some way forward for treatment. And I know you somewhat answered that with seeing if you can transfer the cells into the lungs and human body. But thank you so much for a fascinating conversation. Appreciate your time.
Sure. I mean, I think the short answer for the biomarkers, is we need to develop a nice study, essentially almost like a mini translational study, to see if people that have chronic or acute, response to disease, whether it's COVID-19 or other acute infections, whether there are going to be markers of this cell type, the RAS cell type, that are increased or decreased. Hopefully, increased would be the easiest way to look at these things. And the easiest kind of biomarker would be something that was secreted into the blood. And it's challenging, to do that, obviously, because you got to find the right marker, and it's got to have like enough of a change that you can develop an assay that's sensitive for it. But I think that's coming, and again, I think that would have huge impact to be able to track people that have either chronic or even acute lung disease, whether it's COVID or influenza, and the potential response to treatments. Ultimately, and I'll just sort of end it on this right now too. It's a field, if I can, and get any of you interested in lung biology, it's a field that we have very, very few treatments for. As many of you know, if you go into the hospital, you get steroids, now that we have some antivirals, but you really only have steroids, high flow oxygen, and maybe bronchodilators. We don't have a lot of the medicines or procedures that say the cardiovascular field has. So we've really got to do this and develop some new therapies, and it's critical. We're going to be faced with a tsunami of lung disease in the next 10 to 20 years, and it's really critical that we address some of these things with new therapies.
Yeah, that is actually interesting because we had here a few weeks ago Dr. Cecilia Alham. She actually developed a blood test for Parkinson screening because she works with immune response, especially T cells, that basically recognize the plaques early on and have an increase in a biomarker basically of T cells that already recognize these alpha-synuclein plaques, and yeah, and that way she could develop like an early on diagnosis in peripheral blood for Parkinson. So I was wondering if there's maybe also, would there be a T cell response of when the cells turn into something they shouldn't, or if there's an increase in disease model and some type of signal that the T cells would react to, and that way have a biomarker.
Right, yeah. And I hate to say it, but this will probably be my last answer. I'll have to sign off after this. I have to go to another meeting. But yeah, the immune response is clearly a signal we see in our transcriptomic analysis that these RAS cells, in particular, the ones isolated from COPD patients. So there is definitely changes in the immune response in these cells to this sort of enhanced or increased inflammatory environment in chronic lung disease. So I think that's definitely one way to look at it. Obviously, those inflammatory responses are helpful in many contexts, but they can also be harmful if they are increased for too long a period of time, and they can dampen the ability of the cells either to proliferate or to differentiate. So I'm really sorry I have to leave now, but it's been a wonderful time.
Thank you so much
for your time.
And we really appreciate that you took the time to come here. This is really interesting work, and we wish you all the grant money and
And all the funding, yes, all the
bureaucratic
funding and no
hurdles.
bureaucracy.
That's wonderful. Thank you. I appreciate that. Always a
And,
challenge.
yeah, and hopefully you'll come back sometime with some updates if you would be willing to. Thank you.
Happy to. Thank you very much. Have a good day, everyone. Bye-bye.
Thanks.
Thank you.
Bye.
Have a good day, Doctor. Thank you.
So yeah, thank you everyone for coming. I'm sorry if we didn't get to your questions, but we will hopefully have him back soon at some point. And we usually on Sundays have a weekly recap room where we summarize again the research and give people an opportunity to ask questions maybe they didn't get to ask, and then we try our best to answer them. Or if you have an urgent question, you can message me, and I will then send an email to the guest speaker, and usually they respond. So feel free to message me with any important questions you have, and we'll try to get them answered. And thank you everyone for coming. If you like discussions like these, follow the Science Society. We'll have another room later, and it's the MIT researcher Dr. McGuire. He will talk about newly discovered complex molecules in space that he discovered. It's a really cool, interesting space room later on, and then we'll have a room again on Friday. And I don't know if you know, but our bees, honeybees, are quite struggling, and it's really important that we have them for food security, but also, for diversity of plants, um, because they are pollinators. So we will have a researcher from the UK coming to talk about his newest publication, that he developed a bee that's resistant to some of the diseases that are killing all the bees. So and he's also really nice guest speaker and person. So yeah, feel free to come back, and thank you everyone, and enjoy the rest of your day or evening or morning wherever you are.
Thank you, Katarina.
Thank you for
Okay,
joining us, everybody. Bye bye.
thank you. 3, 2, 1. Bye everyone.